Hello. Today we are going to discuss an important topic titled oxygen dissociation curve. The oxyhemoglobin dissociation curve or oxygen dissociation curve as it is commonly called is vital for comprehending how exactly blood transports and release oxygen. Oxygen is carried through the body primarily by a protein molecule named as hemoglobin, which is present in the red blood cells. We should also know that oxygen can be carried throughout the body in a dissolved state in blood plasma. But this dissolved portion of oxygen constitutes only a small amount of the total oxygen transport in the bloodstream, about 2% of the oxygen in the bloodstream, whereas 98% of the oxygen is bound to hemoglobin. And it is very important to note the vital difference between oxygen saturation, that is SaO2, which is actually a percentage of hemoglobin bound to oxygen. I repeat again, oxygen saturation is a percentage of hemoglobin bound to oxygen, and partial pressure of oxygen in the blood, that is PaO2, which is the amount of oxygen dissolved in the blood. And this oxygen dissociation curve helps us to describe the relationship between these two important concepts. And let us dwell a little bit into the structure of hemoglobin. I request every one of you to read this slide so that you can understand and keep in mind the difference between oxygen saturation and partial pressure of oxygen. I'll use some time for you to read the slide. Of course, you always have the pause button in the video to pause and read, but just I thought I will give you some time deliberately. And hemoglobin as a molecule is a combination of protein globin and a non-protein portion called heme. This molecule is responsible for transporting oxygen in the blood. Hemoglobin, I mean, this here it indicates adult hemoglobin, which is present in adults. So we will dwell a little bit about the type of hemoglobin, which is a little bit different in fetus. That is called fetal hemoglobin. Now I am going to describe the adult hemoglobin. Adult hemoglobin is composed of 4 subunits. That is 2 alpha subunits and 2 beta subunits. And these subunits has a heme group and a globulin chain. I repeat again, each of these subgroups have subunits as heme group and a globulin chain. The heme group is supposed to contain an iron atom in its ferrous form. I repeat again, heme group contains an iron atom in its ferrous form. And then this ferrous form iron is capable of binding one oxygen molecule, allowing one hemoglobin tetramer, that is the 4 components, that is 4 units of hemoglobin, can bind 4 oxygen molecules. I've already told hemoglobin has 4 subunits, and each of these subunits is capable of binding one molecule of hemoglobin. So each of these subunits, when it is bound to one molecule of hemoglobin, totally one hemoglobin molecule can carry 4 oxygen molecules. Hemoglobin physiologically is known to exist in 2 states, that is state T or 10th state, which is in a deoxygenated form where it contains less oxygen, and R state or a resting state known as oxygenated state where it is filled with oxygen. These two states actually differ in their affinity to bind oxygen. I repeat again, these two states differ in their affinity to bind oxygen. That is T state or deoxygenated tense state. And then R state is actually oxygenated relaxed state. I'm repeating again. In an unbound state, hemoglobin exists in the T state. So T state where the oxygen binding occurs with low affinity, wherein it can take more oxygen in a lesser amount of time. The T state hemoglobin thus requires a higher partial pressure of oxygen to facilitate the binding of oxygen. Binding of single oxygen molecule, I repeat again, if one single oxygen molecule binds to the hemoglobin molecule, it opens up the other three sites, whereby other three oxygen molecules can effortlessly bind to the other three sites. So this one binding of one oxygen into hemoglobin facilitates the other subunits into a high affinity R state or relaxed state. The binding is very important. It is important for you to understand the concept of this binding. Then, in a set of normal lungs, the partial pressure of oxygen inside the lungs is supposed to be high at the alveolar capillary junction. Therefore, exposing the tense state or T state hemoglobin to vast amounts of oxygen facilitates oxygen loading into the hemoglobin. This process actually quickly allows the hemoglobin to be saturated to 100% before end of the capillary bed is reached. So the transfusion of oxygen into the hemoglobin happens really fast. On coming out of the pulmonic system or pulmonary system, the hemoglobin is in an oxygenated or R state or relaxed state. However, you should understand the partial pressure of oxygen in the peripheral tissue is supposed to be on the lower side. So, when the hemoglobin, the T state of the hemoglobin or the tense state, T means tense, is actually preferred where the oxygen saturation is low, wherein the hemoglobin in the state, tense state, will release the oxygen quickly. And throughout the bloodstream, different partial pressure of oxygen levels participate in creating an equilibrium between T rates and R rates. Again, I tell you, T is tense state or deoxygenated state. R is actually a relaxed state or an oxygenated state of hemoglobin. The relationship between partial pressure of oxygen and oxygen saturation is classically presented or represented by the oxygen dissociation curve. This is actually a S-shaped curve, and this determines the blood oxygen pressure, the relationship between the blood oxygen pressure to the oxygen carrying capacity of the hemoglobin. So I repeat again in order to avoid confusion. The relationship between partial pressure of oxygen and oxygen saturation is aptly represented by the oxygen dissociation curve. The oxygen saturation is represented in the y-axis as a function of partial pressure of oxygen. And then x-axis, I'm sorry again, I'm confusing you. The curve, if you look carefully, the oxygen saturation is represented in the y-axis. See the diagram, see the curve. The oxygen saturation is represented in the y-axis, and the partial pressure of oxygen is marked in the x-axis. The sigmoid curve or a S-shaped curve is due to the positive cooperative of the hemoglobin. What exactly you mean by the positive cooperativity of the the hemoglobin? in what happens is if one oxygen molecule finds a place inside the hemoglobin, it opens up the other receptors. So in no time, the other three oxygen molecules get bound to the hemoglobin. That is the reason why the curve is pretty steep. And the flat portion of the curve indicates the oxygen level inside the hemoglobin or saturation of oxygen inside the hemoglobin is maintained, and the release of oxygen is really small. Small amounts of oxygen is released in that phase. Moreover, if the hemoglobin is staying in the horizontal phase, oxygen it is in an oxygenated state and it is not in a position to carry any more oxygen. So it has reached a steady state. So horizontal portion of the curve indicates the steady state of oxygenation of the hemoglobin. Then the point where the hemoglobin is 50% saturated is called P50. And this typically occurs at about 26 to 27 millimeters of mercury. Higher P50 or higher P50 means, or higher rate at which 50% saturation occurs means the right shift has occurred, wherein it indicates a hemoglobin has lower affinity for oxygen. So it pushes oxygen outside. Lower P50 means there is a shift to the left, means high affinity to oxygen. It takes in oxygen. I repeat again, there is always a midpoint that is called the point in the curve is 50% of the point, that is midpoint, which happens to indicate the hemoglobin is saturated 50%. It's called P50. This occurs typically at around 26 to 27 millimeters of mercury. Then, when the P50 happens to be at a higher level, it means the shift has occurred to the right. Sorry, if the P50 is at a high level, it indicates the shift has occurred to the right. In other words, the hemoglobin has lower affinity to oxygen. What this causes? This causes release of oxygen into the tissue. A lower P50 indicates the shift of the oxygen dissociation curve to the left. This indicates high affinity of hemoglobin to oxygen. Where this happens? This happens exactly in the lung, where the oxygen from the breeze air gets into the its system by attaching itself to the hemoglobin. Why this curve, oxygen dissociation curve, is S-shaped? The curve is actually S-shaped. I have already told about it. I am just repeating it again in order to reinforce the concept. The curve is S-shaped because hemoglobin shows cooperative binding. How it happens? When one oxygen molecule finds its place, binds to the hemoglobin, the shape of the hemoglobin changes in order to make the other bindings that much easier. So the curve starts to steeply increase. As the hemoglobin reaches the full saturation, when the four slots are filled, there is no sites to be filled by oxygen. The curve flattens at the top. That is actually the horizontal portion of the curve, wherein there is no place in the hemoglobin for oxygen to attach itself. So it is oxygen is not cannot find a place to bind itself into the hemoglobin. So the curve is flat in that area. Now let us discuss delve a little bit into the various key regions of the oxygen dissociation curve. I've already told you the upper area is flat or plateau. This corresponds to the lungs, where the hemoglobin is fully saturated. All the spaces or receptors of oxygen are fully occupied. So there is no more space empty. So the blood cannot carry any more oxygen. Then comes the steep region. The steep region is just below the plateau. This corresponds to the systemic capillaries, where small fall in partial pressure of oxygen releases a huge amount of oxygen from the red cells, red blood corpuscles. So that is actually the steep region. For example, 95 millimeters of mercury, if the with 97% of saturation, is ideal in arterial blood. I repeat again, in 95 millimeters of mercury, that is the pressure, with 97% of saturation, is seen in the arterial blood. Whereas 40 millimeters of mercury with 75% saturation is seen in the venous blood. These two values are sacrosanct and you need to remember it. For example, 95% of millimeters of mercury of oxygen oxygen pressure, 95 millimeters of mercury of oxygen pressure causes 97% saturation in arterial blood. And 40 millimeters of oxygen pressure is there with 75% saturation in venous blood. Now, what happens when the curve shifts to the right? When the oxygen dissociation curve shifts to the right, or P50 shifts to the right, it indicates reduced affinity to oxygen, and it leads to easier unloading of oxygen at the tissue level. This occurs in the presence of carbon dioxide. What happens is carbon dioxide causes increased acidosis, reduces the pH of blood. This leads to the Bohr effect. What Bohr effect says, in the presence of acidosis and in the presence of increased level of carbon dioxide, the oxygen dissociation curve shifts to the right. Repeat again, Bohr effect is again commonly asked short now. So, in the presence of high levels of carbon dioxide, it causes acidosis in the blood by increasing the level of hydrogen ions. This causes a simultaneous increase or shift in the oxygen dissociation curve, causes increased release of oxygen. This is called Bohr effect. The other cause of right shift of oxygen dissociation curve is increased level of 2,3-DPG, diphosphoglycerate in red cell. And right shift of oxygen dissociation curve also occurs during fever. Now, when exactly does the left shift occurs? Increased affinity causes harder or lower downloading of the oxygen. So, when the level of carbon dioxide is less and there is a relative alkalosis in the blood, the shift of the oxygen dissociation curve occurs to the left. Lower temperature again shifts the curve to the left. Carbon monoxide poisoning, what happens is it binds to the tissue, prevents oxygen release, binds to the hemoglobin molecule, prevents the oxygen release, thereby shifting the curve to the left. So carbon monoxide is one of the toxic causes of oxygen dissociation curve shifting to the left. Now let us dwell a little bit into 2,3-diphosphoglycerate in hemoglobin. 2,3-diphosphoglycerate is actually present in the hemoglobin. The presence of 2,3-diphosphoglycerate decreases the affinity of hemoglobin to the oxygen by binding to the central cavity of the deoxyhemoglobin. So it makes it harder for the oxygen to bind, and it is easier, it pushes the oxygen molecule out into the tissues. So the presence of 2,3-diphosphoglycerate pushes the curve to the right, oxygen dissociation curve to the right, because it shifts the curve to the right and increases the delivery of oxygen to the tissues. It is actually a negatively charged molecule and fits into the positively charged central cavity between the beta chains of the deoxyhemoglobin. I repeat again, this 2,3-diphosphoglycerate is negatively charged and it fits into the positively charged central cavity between the beta chains of the deoxyhemoglobin. And this amount of 2,3-diphosphoglycerate rises in chronic or acute hypoxic states as it occurs in mountain climbers, where the shift in the oxygen dissociation curve occurs despite the reduction in the ambient oxygen level. This 2-3, I repeat again, physiologically, the climbers get acclimatized to heights where the lower ambient oxygen levels still help them to sustain is the presence of increased amount of 2,3-diphosphoglycerate, which pushes the oxygen dissociation curve to the right. So the hemoglobin readily gives up oxygen even though the ambient oxygen level is considerably low at altitudes. Now, let us dwell a little bit into the clinical significance. What exactly happens when blood is stored in the blood bank? The 2,3-diphosphoglyceric acid over the time loses its level. So the stored blood loses the 2,3-diphosphoglyceric acid over the time. So what happens is the blood increases, the hemoglobin present in the stored blood has increased oxygen affinity. So imagine you transfuse a stored blood which has been stored for some days into a patient. This blood has, what happens is it has an increased oxygen affinity. So it is not going to immediately provide oxygen to the tissue. Immediately after the transfusion, the oxygen level to the tissue is not going to increase because the stored blood loses 2,3-diphosphoglycerate over the time. The longer the blood is stored, lower is going to be the 2,3-diphosphoglycerate. So what happens? The loss of diphosphoglycerate pushes the curve to the left. When the curve gets pushed to the left, hemoglobin does not readily push out oxygen. So in a patient who receives blood transfusion, there could be a transient problem, but due to this shift of the oxygen dissociation curve to the left. I have been telling about the fetal hemoglobin. Fetal hemoglobin is little bit different than the adult hemoglobin. So what happens is fetal hemoglobin binds 2,3-diphosphoglyceric acid, glycerate, or 2,3-diphosphoglycerate rather poorly. So what happens is in fetal hemoglobin, this 2,3-diphosphoglycerate is bound rather poorly. So the oxygen affinity is very high in fetal hemoglobin. So what happens is this feature helps the fetal hemoglobin to suck oxygen from the maternal blood. So effective maternal to fetal oxygen transfer occurs because the fetal hemoglobin binds 2,3-diphosphoglycerate very, very poorly. So this should be borne in mind. And fetal hemoglobin is different from adult hemoglobin because it has 2 alpha and 2 gamma chains. In comparison, adult hemoglobin has 2 alpha and 2 beta chains. Thank you. Hope this lecture helped you.